Thank you for the introduction. I'm Andalib Nizam. I'm in University of Tennessee PhD student. We work in this project with Oak Ridge National Lab for the whole process. So I'm going to present what we have done with developing the pyrolysis process of these carbon polymer nanoelectrodes. So the outline will be that first I'm going to talk about the motivation behind this project and what previous work has been done by our group. Then I'm going to give you the system overview that we have created. Then I'm going to give a short description of the pyrolysis process and what we have changed or modified during that process. Then the verification of carbonization of these polymers and the biological cell preparation that we have done to create the biosensor prototype, and then the system development and summary. So the motivation behind this project was that we still need carbon nanoelectrodes or nanostructured electrodes for lab-on-chip systems. And the initial efforts that we have done was based on the alignment of carbon nanofibers. However, the integration with CMOS is very difficult in that temperature range, because as you know that pyrolysis of these polymers will happen around 950 to 1000 degrees Celsius, but that is destructive for any kind of CMOS circuitry. So our goal was to decrease that temperature range down to 500 to 600 degrees Celsius, where our CMOS circuit can survive, and we can create these systems in together. So this is the overview of the system that we have created. The metal electrodes are placed on a transparent substrate, usually quartz or silica. Then we develop the 3D printed polymers on top of the edge of the metal electrodes, where you can see that the pillars are grown on top of the metal electrodes, and the bridge structure is the connection between them, so that you can connect your whole circuitry along with that. And there is and the biological cells are then placed on top of that electrode to measure like their cell characteristics. The fabrication process was started with, as I said, that metal electrodes are first patterned on the quartz substrate. And you can see that we used liquid photopolymer, which is IPS and IPDIP. Then we use nanoscrap tool to print the structure with that photoresin, and the process is 2-photon polymerization. Once those are developed, we develop them in SUA developer, and once those are developed, we anneal them in 2 steps. The first step of the annealing is basically a partial carbonization of the polymers, and that happens around 340 degrees Celsius in presence of air or oxygen. The temperature usually rises from 40 degrees Celsius up to 340 degrees Celsius in about 10 minutes. Then we keep that temperature constant for another 10 minutes, and then it cools down to 340 degree down to room temperature. Then we place this sample in a rapid thermal processor unit, where it is annealed at 600 degrees Celsius for 10 hours in presence of argon and at a pressure of 9.9 torr. So these are some of the samples that we have developed, and we have repeated that process with different laser power in the input, different percentage of laser dosage. As you can see in the ACM image, the initial size of the bridge was 4 micron in diameter, but it shrunk down to about 70 to 100 nanometer, which is a common phenomenon that whenever you are burning polymer or pyrolysis, it will start to shrinkage, will be affected by shrinkage. So one of the sample here is developed with IPDIP, and we have seen similar performance as to as compared to IPS. So some of the modifications we have to make, because we have in our group, we have established this recipe, and what we have seen that if your IPS photoresin is not stable or it is close to aging, then you may have different results where your structures may destroy. And also the laser power input of the nanoscrap tool that needs to be well calibrated to generate these kind of good structures. In place of that, if you increase the laser dose, that will often give you better results after the pyrolysis. But the problem with that is it will, once you increase the input laser, then your voxel size will increase, which results in less resolution in your structure. And the use of IPDIP was another comparison we wanted to try with compared to IPS. We have seen that it has reducing aging effect for the same batch of that was purchased for IPS and IPDIP. And it is easier to carbonize with shorter duration of the high anneal time. But the problem is that it is much less smooth than IPS structures. So it can also have inconsistent sizing between your structures. So here is an example of two samples developed, one in 2022 and another in 2023. And it also shows that this process is very reproducible, and this recipe it works over time with different samples with different type of photoresin. Next, the verification of the nanoelectrodes, we verified with the Raman spectroscopy. As you can see that there are two peaks in the Raman D band and G band, which basically shows that it has sp2 structures in carbon presence in the structures. So we also did Raman for pillars and bridges together to see whether they have the same kind of profile in Raman spectroscopy. And this also verifies the SEM image observation that there is carbonization. So the next step was to apply biological cells on top of these electrodes and measure the cell impedance as a first test. So the cells are derived from glioblastoma cells that was cultured by another department in our university, Center for Evolutionary Biochemistry. And the cells were then placed with a media present on them. And we developed two setups where for the first setup, the metal electrodes that we connect, there is a bridge and pillar structure vertically between them. And for the second setup, there is no bridge and pillar structure, only the cell and the media present between the metal electrodes. So as we have seen that with only media present, the impedance remains constant. That is the baseline of these experiments. And then as we apply cells in there, we get two different plots of the impedance increasing as the cells settle down and starts multiplying over time. So once it settles down and it starts to grow, you can see over an 8-hour period of time, there is an increase of impedance. So to summarize the project, the fabrication of the carbon nanoelectrodes typically take place at higher temperature, but we have achieved successful carbonization in a much lower temperature that is compatible with CMOS circuitry. The photoresin used and the laser properties that significantly can affect these structures, we have demonstrated that it can be balanced with other approaches. And then we have verified the structural stability and carbonization with Raman spectroscopy and SEM imaging. And then the fabricated structures were placed with the cells to measure the impedance as a first run. And future work will be involving to separate and isolate the electrodes with the cells. As you can see that it was all over the electrodes. So we want to do a much controlled experiment with that. That's all. And I want to lastly acknowledge Oak Ridge National Laboratory for the fabrication and characterization of these samples, and Dr. Muhammad Aminul Haq, who is to be in our lab for the figures in slides 3, 5, and 6. Thank you.